Welcome to the chapter on nervous system and senses. This chapter is quite complicated. However, I'll try to focus on some of the important things that you need to know at this level. Nervous system is the most complex system in our body, and it has a long list of functions. However, we can divide the overall function of the nervous system into three categories. They include sensory function, integrative function, and motor function. Under sensory function, our nervous system receives the sensory information from receptors and carry it to the brain and spinal cord. Under integrative function, our brain and spinal cord process the sensory information and try to make sense out of it. And then under motor function, the nervous system sends impulses to our effector organs. Effector organs are usually muscles or glands. Organs of the nervous system can be divided into two groups: central nervous system, or CNS, which is made up of the brain and spinal cord, and peripheral nervous system, or PNS, which is made of the nerves coming out of the brain called cranial nerves and the nerves coming out of the spinal cord called spinal nerves. Information from sensory receptors get to the central nervous system through sensory division of peripheral nervous system. This information is processed in the central nervous system, and based on that, a decision or a command is sent. The command is sent in the form of electrical impulses through motor division of peripheral nervous system to various effector organs. Motor division is further divided into two groups. If the motor division ends to a conscious organ such as skeletal muscle, it is called somatic nervous system. However, if the motor division ends to an unconscious organ such as smooth muscle or cardiac muscle, then it is called autonomic nervous system. The nervous system is made of two types of cells, neurons and neuroglia. Neurons are the type of cells which has the ability to conduct electrical impulses. And one neuron communicates with another neuron through chemicals called neurotransmitters. On the other hand, we have cells which has nothing to do with conduction of electrical impulses, but they provide more of a supportive role, and they nourish neurons and protect neurons. These cells are called neuroglia. This slide shows an image of a typical neuron. A neuron cell is made of three parts. First of all, you have the soma, which is also called cell body. There you find the nucleus and other organelles such as Golgi apparatus, mitochondria, and what have you. From soma, you have several small extensions. These small extensions are called dendrites. And then from soma, you also have one large extension, which is called axon. At the area where axon originates is called axon hillock. So the direction of information is that the information is received by dendrites, then they get to soma or cell body, and then they move along the length of axon. The axon in some part of the nervous system is wrapped up by sheets of cells. These sheets of cells are called a myelin sheet. And the purpose of myelin sheet is to speed up the electrical impulses. So once electrical impulses travel along the length of axon, they get to the end part of the axon, which is called axon terminal. You can probably look at this image that the end part of axon is slightly bulged. That is called the synaptic knob, where you have the neurotransmitters stored. So when the electrical impulses get to the axon terminal, neurotransmitter is released, and then the neurotransmitter will go ahead and stimulate the following structure. It can be a neuron or it can be a muscle. In terms of functional classification of neurons, we have three types of neurons. So first of all, we have sensory neurons. Sensory neurons are also called afferent neuron. Their function is to carry information from sensory receptors to central nervous system. The second type of neurons is called interneuron. Interneurons are found in the central nervous system, and their function is to transfer or communicate information from one part of the central nervous system to another part of the central nervous system. And finally, we have motor neurons. Motor neurons are also called efferent neurons. Their function is to carry electrical impulses from central nervous system to effector organs. So as I said earlier, that neuroglia are the types of cells which support neurons and they protect neurons, but they have nothing to do with conduction of electrical impulses. There are different types of neuroglia, both in the central nervous system and in the peripheral nervous system. The most important neuroglia cell in central nervous system include microglia. And microglia are basically macrophages in the nervous system. Their function is to engulf and eat and break down things that are not needed anymore. This can include a bacteria, a faulty protein, a dead cell, what have you. Then we have oligodendrocytes, and the function of oligodendrocytes is to produce or form myelin sheet around the axons in the brain or spinal cord. Ependymal cells produce a fluid which showers the central nervous system, nurture the central nervous system, and also carry electrolytes from one part to another part, which is called cerebrospinal fluid. Finally, we have astrocytes, probably one of the most important neuroglia in the central nervous system. Their function is to provide structural support, but I think the most important function of neuroglia of astrocyte is formation of blood-brain barrier. They protect the blood vessels in the brain and making sure that not all the chemicals leave the blood vessel and get into the brain because it may alter the brain function. There are two types of neuroglia in peripheral nervous system, but at this level, let's focus only on one type, and that's called Schwann cell. And similar to oligodendrocyte, the function of Schwann cell is to form myelin sheet around axons in the peripheral nervous system.
The human brain alone contains about 100 billion nerve cells called neurons. A neuron, like all cells, has negatively and positively charged ions in the fluids located both inside and outside of the cell. However, the ionic composition of these two fluids is markedly different. The inner surface of the membrane is negatively charged, while the outer surface is positively charged. This unequal distribution of charge creates a voltage across the plasma membrane, known as the membrane potential. In a resting, unstimulated neuron, this membrane potential is known specifically as the resting membrane potential. The value of the resting membrane potential depends on two factors. The first is the presence of concentration gradients across the plasma membrane, especially those gradients for sodium and potassium ions. The second is the permeability of the plasma membrane to sodium and potassium. In the resting state, this permeability depends on the number of non-gated leak channels present in the membrane for these ions. The magnitude of the resting membrane potential, inside relative to outside, is measured in millivolts using a voltmeter and two electrodes. We'll now place both electrodes in the extracellular fluid. Note that the voltmeter records 0 voltage. This is because the unequal distribution of charge exists only across the plasma membrane. The intra- and extracellular fluids are otherwise electrically neutral. However, when we insert one of the electrodes into the intracellular fluid, a voltage is recorded. The value of the resting membrane potential varies, but on average it is -70 millivolts. There are two major fluid compartments in the human body. Extracellular fluid surrounds our many trillions of cells, and intracellular fluid resides within those cells. Although these fluids consist mainly of water, they have very different ionic compositions. The extracellular fluid has higher concentrations of sodium and chloride ions, while the intracellular fluid has higher concentrations of potassium and negatively charged protein ions. Note that the large size of these protein anions makes them impermeable to the plasma membrane. The concentration or chemical gradients for sodium and potassium are similar in size, but they drive diffusion in opposite directions. Sodium diffuses to the inside of the cell, as potassium diffuses to the outside. As you have seen, concentration gradients drive sodium ions into the cell and potassium ions out of the cell. However, potassium diffuses more easily because the membrane has many more leak channels for potassium, making the cell more permeable to potassium. Due to this greater permeability, the resting membrane potential is largely determined by potassium diffusion. So for simplicity, let's begin by imagining a simplified neuron, one whose membrane is permeable only to potassium. This will let us focus on the effect of the potassium concentration gradient. To explain the generation of the resting membrane potential, we're going to temporarily set the potential to 0 and then observe what happens as it polarizes to its negative resting value. Positively charged potassium ions diffuse down their concentration gradient to the outside. This causes positive charge to accumulate along the outer surface of the plasma membrane. The departing potassium leaves behind negatively charged ions, most notably the large protein anions, which are impermeable to the plasma membrane. As a result, the inner surface becomes negatively charged. This separation of charge gives rise to a membrane potential. The membrane potential's value depends on the net charge inside compared to outside. As positively charged potassium ions continue to diffuse out of the cell, the membrane potential becomes increasingly negative. As we've learned, when potassium diffuses down its concentration gradient to the outside, the inside surface of the plasma membrane becomes negatively charged. Observe the membrane potential becoming increasingly negative as potassium ions continue to diffuse out of the cell, down their concentration or chemical gradient. However, as you'll see in this animation, the factors influencing potassium diffusion are a bit more complex. As potassium leaves the cell, the negative charge accumulating on the inner surface of the membrane establishes an electrical gradient that draws positively charged potassium ions back into the neuron. Thus, the overall direction and magnitude of potassium diffusion depends both on its concentration gradient and its electrical gradient. The combined effect of the two is often referred to as an electrochemical gradient. As more potassium diffuses out of the cell, an increasingly negative membrane potential develops. This increases the electrical gradient on potassium, opposing further diffusion of potassium to the outside. The membrane will eventually reach a potential at which there is no net driving force pushing potassium in either direction because the electrical gradient exactly opposes the concentration gradient. In our simplified neuron, which is only permeable to potassium, this occurs at a value of -90 millivolts. At this potential, there is no net movement of potassium, as movement is equal in both directions. When we considered the effect of concentration gradients on the membrane potential, we used a simplified neuron whose membrane was permeable only to potassium. We learned that at a membrane potential of -90 millivolts, there is no net electrochemical gradient acting on potassium, because the chemical gradient driving potassium out exactly opposes the electrical gradient driving potassium back in. Under these conditions, there is no net movement of potassium. We'll now consider a real neuron, one whose membrane is not only very permeable to potassium, but also slightly permeable to sodium. How does sodium permeability affect the membrane potential? Recall that the concentration or chemical gradient for sodium drives it into the cell. In addition, the negative membrane potential creates an electrical gradient that also drives positively charged sodium into the cell. The combined electrochemical gradient for sodium is therefore large and directed inward. The entry of positively charged sodium ions makes the membrane potential less negative, but because sodium permeability is low, the effect is small. In a real cell, the membrane potential eventually stabilizes to about -70 millivolts, which is the resting membrane potential of a typical neuron. At this voltage, even though there is net movement of both sodium and potassium ions, the membrane potential remains steady. This is the resting membrane potential of a typical neuron. The resting membrane potential thus depends not only on the concentration gradients for sodium and potassium, but also on the relative permeability of the plasma membrane to these ions. At the typical resting membrane potential of -70 millivolts, the chemical gradient pushing potassium out of the cell is slightly larger than the electrical gradient pulling it back in, so a small amount of potassium continuously leaks out. At the same time, a small amount of sodium continuously diffuses in. The chemical and electrical gradients for sodium both force it into the cell, but because the membrane is only slightly permeable to sodium, only a small amount leaks in. So even when the neuron is at rest, potassium constantly leaks out and sodium constantly leaks in. Just as a boat that springs a leak will eventually sink, a leaking neuron will eventually fail to function. The leaks will eventually change the ion concentrations enough that the neuron cannot generate the electrical signals it uses to communicate. To compensate for a leaking boat, the captain can bail water out as fast as it enters. Neurons also compensate for their resting leaks by transporting potassium back into the cell and sodium back out of the cell. This is done by sodium-potassium pumps in the membrane, which actively transports sodium and potassium ions against their electrochemical gradients. The pump is important for the resting membrane potential, but its effect is largely indirect. It maintains the concentration gradients that determine the resting membrane potential. Let's review what you've learned. The term membrane potential refers to the voltage created by a separation of electric charge across the plasma membrane. In an unstimulated cell, this voltage is called the resting membrane potential. Concentration gradients and membrane permeability drive the differential diffusion of sodium and potassium ions across the membrane. The diffusion through leak channels in a resting neuron determines the magnitude of the resting membrane potential. In a typical neuron, the value of the resting membrane potential is -70 millivolts. At this voltage, sodium leaks into the neuron and potassium leaks out. These leaks are counterbalanced by the action of the sodium-potassium pump, and the potential remains steady at -70 millivolts. Neurons communicate over long distances by generating and sending electrical signals called action potentials. Most action potentials originate in the axon's initial segment, a region also called the trigger zone. During an action potential, voltage-gated sodium and potassium channels open and close, thus altering the membrane's permeability to sodium and potassium ions. As these ions diffuse through the channels, the membrane potential changes. It depolarizes from the resting state of -70 millivolts to a peak value of +30 millivolts, and then quickly repolarizes back to -70 millivolts after a brief period of hyperpolarization. In this topic, we explore, step by step, the events that occur as an action potential is generated in the axon's initial segment. The action potential begins when local signals originating from the dendrites and cell body reach the axon's initial segment. These local signals, also called graded potentials, are very different from action potentials in that they are short-lived and their magnitude decreases with distance. However, when their magnitude is great enough, they produce currents that spread all the way to the initial segment. When this occurs, it causes the membrane potential to depolarize to a critical voltage called threshold. Threshold is the membrane potential that must be reached in order to generate an action potential. Its value is about -55 millivolts. The membrane depolarizes further to a peak value of +30 millivolts. The membrane then repolarizes and returns to -70 millivolts, following a brief period when the membrane potential becomes more negative than the resting state. This period is known as hyperpolarization. If the currents arriving at the initial segment are insufficient, and the change to the membrane potential is below -55 millivolts or subthreshold, then no action potential fires. In fact, action potential generation is an all or none event. Either it happens or it doesn't. When the initial segment depolarizes to a suprathreshold value, even one that is way past threshold, an action potential of the same amplitude is produced. Action potentials are always the same. The amplitude never varies. The sodium and potassium ion channels involved in the generation of an action potential open and close in response to changes to the membrane potential, or voltage. Thus, these channels are voltage-gated. The voltage-gated sodium channel has two distinct gates. One is an activation gate. The other is an inactivation gate. The two gates open and close, alternating between three different states. The first state is the closed state that we observe at the resting membrane potential. The inactivation gate is open, but the activation gate is closed. No sodium ions can pass through the channel. The second is an open state. With sufficient depolarization, the activation gate opens, permitting sodium ions to pass through the channel. The third is an inactivated state, observed during repolarization. The activation gate remains open, but the inactivation gate closes. This blocks sodium ion diffusion once again. In order for the channel to reopen, it must first return to its closed state. The voltage-gated potassium ion channel is simpler because it has no inactivation gate. It has a single activation gate that alternates between being open or closed. At the resting membrane potential, the activation gate is closed. With sufficient depolarization of the membrane, the activation gate opens, allowing potassium ions to pass through the channel. Now let's compare the two channels side by side. The sodium ion channel has two gates. One is an activation gate, and the other is an inactivation gate. In contrast, the potassium ion channel has only a single activation gate. Both channels are closed when their activation gates block the diffusion of their respective ions. Note that in this closed state, the sodium ion channel's inactivation gate remains open. Both channels permit the passage of their respective ions when their activation gates swing open. In this open state, the sodium ion channel's inactivation gate remains open. As stated before, the sodium ion channel has an additional state that we don't observe in the potassium ion channel. This is an inactivated state in which the activation gate remains open, but the inactivation gate closes. Let's examine the phases of an action potential. We start at the neuron's resting state. A threshold stimulus opens both voltage-gated sodium and potassium ion channels in the neuron's membrane. However, these two types of channels open at quite different rates. Sodium ion channels are quick to open in response to the stimulus, while potassium ion channels are slower to open in response to the same stimulus. Let's take a closer look at this again. As the activation gates of some voltage-gated sodium ion channels open, sodium ions diffuse into the cell. Sodium ion entry depolarizes the membrane further, which opens more sodium ion channels, leading to even greater sodium ion influx. This establishes a positive feedback loop that switches on as the membrane potential passes threshold. Depolarization of the membrane opens sodium ion channels. Sodium ions enter the initial segment and depolarize it further. As more sodium ion channels open, we observe a rapid depolarization to a peak value of plus 30 millivolts. This constitutes the depolarization or rising phase of the action potential. An abrupt decline in the membrane's permeability to sodium and an increase in its permeability to potassium mark the end of depolarization and the beginning of the repolarization phase. Voltage-gated sodium ion channels, which began to inactivate late in the depolarization phase, continue to close their inactivation gates. Voltage-gated potassium ion channels, which began opening during the depolarization phase, continue to open. At the peak of depolarization, more potassium ions are leaving the neuron than sodium ions are entering, and the membrane potential begins its return to a negative value. We now enter the repolarization or falling phase of the action potential. Notice that potassium ion diffusion out of the neuron continues beyond the point of restoring the membrane potential to its resting state. The membrane potential actually hyperpolarizes to about minus 80 millivolts, thus producing the hyperpolarization phase of the action potential. Ultimately, potassium ion channels close and the membrane returns to its resting state. By this point, all voltage-gated sodium ion channels have reset to their closed state. This means their inactivation gates are open and their activation gates are closed. A common misconception about this process is that a huge number of sodium and potassium ions must cross the membrane to produce an action potential. In reality, relatively few ions move, and the concentration gradients between the inside and outside of the neuron remain essentially unchanged. However, the sodium and potassium ion gradients can be disrupted after the repeated firing of thousands of action potentials. Over the long term, neurons rely on sodium-potassium pumps to maintain these gradients. Let's review what you have learned about the generation of an action potential. Voltage-gated sodium and potassium ion channels are closed in a neuron at rest. A stimulus that reaches threshold slightly depolarizes the membrane of the initial segment, or trigger zone, where the action potential originates. At this point, some voltage-gated sodium channels open, allowing sodium ions to diffuse inward. The membrane depolarizes more, and a positive feedback loop is initiated that opens all voltage-gated sodium ion channels in the initial segment. During depolarization, the membrane potential surges to plus 30 millivolts. And it is during this phase that voltage-gated potassium channels begin to open. At the end of the depolarization phase, voltage-gated sodium ion channels inactivate, as more voltage-gated potassium channels open. Potassium ions then diffuse out of the initial segment, and the membrane repolarizes back to a negative value. Voltage-gated potassium ion channels remain open after the membrane potential reaches its resting state, and the initial segment hyperpolarizes. As the membrane potential returns to the resting state of minus 70 millivolts, all voltage-gated sodium and potassium ion channels revert to their closed states. Over time, the intracellular and extracellular concentrations of sodium and potassium ions are maintained by sodium-potassium pumps.